Bonjour tout le monde. Thank you for coming. Wonderful to see some familiar faces here and also some not familiar faces. Welcome everybody. So yes, it's a huge pleasure and an honor to introduce Steve Quake, who's coming here to give 4 lectures at the Collège. As you know, these are series that we do for exceptional people, and I think Steve, we can safely say, is exceptional in many, many ways. So I'm going to give a bit of an intro. So Steve's actually a physicist by training. He got his first degrees in physics and maths from Stanford in 1991, and then he went on to do a PhD in theoretical physics at Oxford in 1994 as a Marshall Scholar. And his thesis research was actually on statistical mechanics and the effects of knots on polymers, even before they became fashionable. But they're the key to understanding a lot. Anyway, so he did his postdoc at Stanford in the lab of Stephen Chu, another great scientist, single molecule biophysics postdoc. And at a very young age, he was already hired at Caltech to become faculty at the age of 26. He then went on to be the Thomas and Doris Everhard Professor of Applied Physics and Physics. So as you can see, very much a physicist doing biology, but a physicist who loves biology. And then he moved back to Stanford in 2005, where he helped to set up a brand new department in bioengineering. And he's currently the Lee Otterson Professor of Bioengineering and Applied Physics. And from 2006 to 2016, he was also an HHMI, Howard Hughes Medical Institute investigator. He has won so many awards, I won't even begin to tell you, for his contributions to science, many, many. And he's one of the few, I think it's 24 scientists who's been elected to all 3 of the US National Academies. So Steve, I had written down, was currently the head of science at the Zuckerberg initiative, but he actually told me he just stepped down. It was announced a couple of weeks ago, which I didn't realize. So congratulations, Steve. I think I can imagine that it must be wonderful to have a bit of freedom. But while you were there, you were transformational. You were in charge of all the science grant programs, technology development, and you were actually the founding co-president of the Chan Zuckerberg BioHab initiative between 2016 and 2022. And you've continued your science throughout all the big things that you've taken on. And I think we can safely say you're not just a deep, brilliant scientist, you're a real innovator and an entrepreneur. And so you're very well known for the many technologies and approaches that you've brought to biology with your physics and engineering background. So approaches that allow us to make accurate biological measurements and he helps us think about the good questions to ask that could be measured. So you contributed to the field of microfluidics in particular, including microfluidic large-scale integration. You developed many of the applications of microfluidics that we see today, drug discovery, also in structural biology approaches, molecular affinity measurements. And you've also been a big player in the field of genomics, and in particular, the whole era of single molecule DNA sequencing, and also the platforms and techniques needed both to do and to analyze single-cell gene expression data. So the Human Cell Atlas and other cell atlases are very much also thanks to your input and your initiative. So a lot of what you do is extremely useful to basic research and basic science, but also very quickly applied to medical applications. So many of the tools you've made for non-invasive prenatal diagnostics, instead of amniocentesis, non-invasive tests for heart transplant rejection, ways of looking at the immune system. So, you know, really a very wide, broad spectrum of different things that you do. And yet you have time to be, I would call you one of the scientists who's a true philosopher. You have time to sit down and think with others, and you enjoy it. You enjoy communicating with others. And so I feel like we're very lucky to have you here. at the Collège for this series of lectures. We're very happy that you appreciate Europe as much as you do. And even more so, we're very lucky to have people like you in science, given what's going on. So it's a real pleasure to have you here. And I will now hand over to you, but just say that these 4 lectures are going to be around the topic of the sort of fundamental importance of cellular biology and medical applications. And in this lecture, we'll be diving into the mysteries of the cell. So thank you so much, Steve. Over to you.
Well, thank you so much for inviting me here to the Collège. It's a real privilege to be here. And I was going to try to give the lecture in French, but maybe not the first one. Maybe I'll get my courage up over time. So this will start with sort of very basic general introduction and then get more and more detail as we go on. And so if the introduction is something very familiar to you, just remember what the great author Mark Twain said: there's nothing ever so pleasant as thinking about something you already know. So we'll start with the mystery of life. And it was sort of the secret of life. It was famously termed so by Watson and Crick, which is the DNA molecule. It's the molecule which is the tool of genetic inheritance in all of our cells. The structure was solved in part due to contributions from Paris here, which I'll talk about in a minute, half century ago. And in solving the structure of the molecule, the method by which it stores the information became apparent. And so if you look at it, let's see, is there a... Maybe not. So if you look at the structure of the molecule, it's famously a double helix, which is a ladder as if it was twisted around.
Oh, yeah, it's really not.
Yeah. It's okay. We'll make it. And the chemical rungs of the ladder are the 4 famous base pairs, shortened as A, C, T, and G: adenine, thymine, guanine, and cytosine. And they encode the information for how to make proteins and perhaps indeed how to make the whole organism. There we go. Merci beaucoup. So the 4 letters shown here, A, T, G, C, these are the rungs of the ladder. There's sort of a redundant coding in the sense that there's a mirror image of how information is encoded. Also this suggests how the cells might divide and how the molecule is replicated. And the history of this is written in sort of, I would say, 3 canonical books if you're interested in the story. Both Watson and Crick wrote their sort of personal memoirs of how they arose at the discovery. And the key person who was left out of the prize giving in this event, but actually did the experiment and acquired the data that allowed the solving of the structure, was Rosalind Franklin. And her story has been recovered in history. And this is a lovely biography of her contributions. And she actually trained here in Paris. She learned how to do crystallography under Jacques Mering, and she brought that back to the UK, where she began working on the structure of DNA. And so Paris played an important role in the whole story of the secret of life. So just to give you a sense of how unusual a molecule DNA is, let me take you through some of the numbers. Every one of our cells has our entire genome in it, almost, a few small exceptions. And if you were to take the DNA in your cell and stretch it out end to end, it would be 2 meters long, so about that long. Every single one of your cells has a molecule that is this long. And we all have about 30 trillion cells in our body. And so if you took all the DNA in your body and stretched it out end to end, it would go all the way from the Earth to the sun and back 250 times. It's amazing. How does that all fit into such a small space? That's a very interesting question. The answer is, even though it's long in one dimension, it's very small in the other 2 dimensions. So it's atomic in dimension, just a few atoms wide in the other 2 dimensions. And that is how you can sort of make it fit into such small spaces. And because it's long in one dimension, it's so long you can actually see it. This is a movie we made in my lab some years ago of a DNA molecule, which is the long piece of string there, being tied into a knot. As Edith mentioned, that was one of my passions early on. And this is a very complicated knot. It's crossing over itself something like 17 times. But it's amazingly fascinating to me that you can look at molecules. You could see them with your eyes if you wanted through the microscope. In this case, it was taking the movie with a camera. So that is phenomenal. And DNA was the first secret of life. The second secret of life was discovered here in Paris at the Pasteur by Jacob and Monod. There's sort of two second secrets of life, but the one I want to focus on here is this notion that some genes control other genes. Okay? So if the DNA has the instructions for all the genes and how to make the proteins associated with them, how does it all get controlled? And why aren't all genes made at once at the same time? And it has to do with this phenomenon called regulation that was discovered here. Further work established that the way regulation works is some proteins will bind to the genome and control whether genes are copied into RNA or not, turned on or turned off. Here's sort of a schematic diagram in the case of higher organisms. Here's the genome. Here's the first transcription factor. Here's other proteins that will bind to that and help the cell decide whether or not the RNA polymerase can come in and effectively turn the gene on and make its product. So that ends up being very important. And these molecules that control whether genes are turned on or not end up being very central to what the cell does and are going to be an important focus of the lecture today. So the so-called central dogma, how this all sort of works, is DNA is like the library. It's got a list of all the genes, which are the instructions for how to make proteins. Based on this control, this regulation, some of them will be transcribed, copied into RNA, then the RNA is translated into proteins. Then some of these proteins in turn will come back and regulate the DNA. If you'd like to look at the story of how all this was worked out, there's a lovely history by Horace Freeland Judson called The Eighth Day of Creation. And it is just a beautiful history of the whole field, the early days of molecular biology. And Monod had this famous quote, one of many, "Anything true of E. coli must be true of elephants." All this work was initially discovered in this humble bacteria called E. coli. And the idea is many of the things discovered, many of the principles would generalize to other organisms, indeed all organisms. Some of this is very universal. And that is something we'll come back to as well during the course of the talk. So where are we now with the genome? The genome has been sequenced. 20 years ago, the human genome was sequenced, and something like 30 years ago, the first bacterial genome was sequenced. And yet, biology is not finished yet. The genome isn't the answer to everything. It's often been called the blueprint of the organism, but I really think that's a misnomer. It's more like a parts list. One way to understand that, and which is going to be the theme of the lecture today, is different cells in your body all have the same genome, but they end up being very different. A neuron is very different than a heart cell, is very different than a lung cell. They look different, they do different things, but they all have the same genome. How does that happen? How does that work? Nobody has been able to predict those cell types from the genome. That's really, to my mind, a tremendous open question for the field. If you just take a new organism that had never been studied, sequence its genome, it's impossible to predict what are the various cell types in that organism just from the knowledge of the genome alone. And there's many other interesting questions, open problems, and how you can interpret the information stored in the genome. It's really, having the sequence doesn't mean we understand what it does by any means. There's much work left to do. And so, if we can't predict or compute the cell types of the organism from the genome, how do we get them? So far, the answer has been to measure them experimentally. And that's what I'm going to spend the rest of the talk discussing, is how we set about on a decade-long program, actually 33-decade-long program if you consider the whole field, but the part I was involved in, of building cell atlases of all the cell types of the human body and of many other organisms. And we went about that using a variety of very neat technologies, and I'll spend a few minutes now telling you about some of those technologies. Set up a new institute in San Francisco. This was the BioHub. We sort of set it up to do this as a large project. But it builds on quite a bit of work that was done over 30 years. the ability to sequence all the RNA molecules in the cell is kind of the key approach to this characterizing all the cell types. Because if you measure the RNA, you know which genes are turned on in a given cell at a given point in time. And so that ends up being the important thing to measure. And people began thinking about that back in the 1990s and figuring out ways to biochemically amplify all the RNA from single cells. There were 2 papers in 1990 independently by Eberwein and Iskov who had used the just then recently invented method of polymerase chain reaction, which is the first biochemical amplifier, to amplify the contents of single cells. But they couldn't read out all the results. It was so complicated, there was no easy way to read out the complexity of all the RNA they had amplified. And that required developing a new technology, which initially was done through what's called microarrays. And in 2003, 4 papers were published showing how to do that. And then ultimately by directly sequencing the products. Sequencing technologies have become very sophisticated and inexpensive to do. And in the late 2000s, the first single-cell sequencing was done. And this allowed one for the first time to really probe the full complexity of gene expression in a single cell. The challenge, and the piece that was left undone up to that point, was how to do that on lots of cells. And these experiments were all done, they were very bespoke, done with a handful of cells, and it wasn't something that could be done at scale where you could contemplate doing it over hundreds, thousands, or millions of cells. And that's where I became involved with the field. My research at that point was involved in trying to make small plumbing to automate biology. We thought about that as the integrated circuit of biology, but instead of having transistors and logic gates, these chips had pipes and valves and pumps on them, and we figured out ways to build chips that had tens of thousands, even hundreds of thousands of valves on them. This is a picture of one. It's an actual picture. The channels here were all filled with food coloring, so you can get an idea that the complexity of the plumbing approaches that of an integrated circuit. And we use these chips in this approach to look at many different fun problems in biology, and ultimately realized we should use them to do single-cell genomics. And a company I founded called Fluidigm built the first commercial product to do this. It was called the C1, and it first came on the market in 2012. And it sort of opened up the field for any biologist to be able to use this approach and these tools to probe biological complexity. And figuring out how to build that plumbing was a whole story in and of itself. And the company eventually produced billions of valves, which is kind of amazing. And these chips allowed biologists to do 100 single-cell experiments at a time. 100 single cells could be measured at one time. It was amazing. And hundreds of labs adopted it, thousands of researchers, many beautiful papers. But that only whetted people's appetite for more and more such data. And eventually, a second generation emerged, again based on technology developed in my lab around microfluidics, in this case, miniaturized droplets. And a group of faculty in Boston first applied this approach to single-cell genomics, and that's now commercially available. It allows thousands of cells to be measured at a time, and now we're seeing a third generation of technology emerge into the field, which allows one to measure hundreds of thousands to millions of cells. So it's been an amazing trajectory. Costs have dropped by more than a factor of 1,000. Oh, and I should say that the story of microfluidics also has an interesting chapter here in Paris. The Institut Pierre-Gilles de Gennes, right down the street, has some of the world's leaders in this field and have made many important contributions. So let me sort of summarize how we go about making these cell atlases and do these experiments. The idea is you take a tissue and you cut it out of its donor. You chemically dissolve the bonds between the cells, so the individual cells are there in solution, separated. Then you use these microfluidic tools in order to separate the individual cells, break them open, amplify their RNA, and basically convert it into something that will be read out by the sequencer. That material is loaded onto a sequencer, it's sequenced, and what you get out for every single cell is essentially 20,000 measurements. Okay? In the human genome, there's roughly 20,000 genes, and so for every cell, you learn how many mRNA molecules from each gene was being expressed in that cell. It's a 20,000 dimensional vector. It's a very sort of complicated, information-rich result, and then you get to compute on that and get to try to understand what it's telling you. You'll see some figures like this later in the talk, and just to help you interpret them, every single dot is a single cell. That's 20,000 dimensions. It's been projected into 2 dimensions here, and the colors represent what's close together in the 20,000 dimensional space, and there's a lot of interesting math for how that's done. So we'd spent, in my lab maybe 10 years working out this part of the puzzle. This part of the puzzle had, the amplification part had been worked out much earlier, as I mentioned. The sequencing part had been worked out. There's a very interesting story behind that. And then about 10 years ago, we set out trying to do this at scale and do a very large project that would allow us to characterize all the cell types, not just in the human body, but also in mouse and in fly and in lemur. We're able to do this as part of a very large team science approach, combining the efforts not just of the researchers at the BioHub, but also at our partner universities, Stanford, UCSF, and Berkeley. These papers had like 150 authors on them. It was really a very different sort of science than anything I had done before, but was necessary in order to take on a project of this scale. And now let me tell you a little bit about some of the stories that came out of that. The first one we did was mouse, and that's a great model organism. We decided to work out sort of the process of how we do this in mouse. And what we decided to do, we were looking at something like 18 different tissues. We decided we'd try to get them all from the same mouse. And the reason for that is we wanted to study aging in mouse and how the cells were changing with age. And we had a collaborator named Tony Wiscarret who was able to get very old mouse. 30-month-old mice is roughly the same as 100-year-old humans. And so it's a chance to look at sort of extreme aging, but not many mice live that long, so there weren't a lot to work with. So we decided we have to try to get the most we could out of each mouse. So in the summer of 2017, pretty much every morning, this group of students and postdocs showed up in Tony's lab with their ice buckets, and each one took a different tissue from the mouse back to their lab to begin to process it. And each of them came from a lab that was an expert in that particular tissue. So we had lung experts, liver experts, and so forth and so on. And we learned how to do the teamwork around that based on mouse. What came out at the end of the day was a ton of data. And these experts would sit down and analyze the data from their tissue and label each cell based on what cell type they thought it was, based on the genes that were expressed there. And then we began to ask the computer to work on the data. And we didn't tell the computer what the humans had worked out. The computer got only the numbers and didn't know any biology. And we asked it, which cells are like each other, which are different? If you cluster them together in the high dimensional space, what happens? This is what happens for the 3-month mouse. And each of these colors is a different tissue. And the labels here are what we added later based on what the humans had done in their annotation. What you see is, based just on those numbers, those 20,000 numbers for each cell, there's a lot of information there. And in fact, cells of different tissues will cluster together. They're similar enough, the computer recognizes that. And so you can see from the clusters with pure colors, those are certain cell types. Some clusters have mixed colors in them. And those are very interesting. Those are cell types that are found in multiple tissues. Okay? So certain immune cells, the B cells which make antibodies, are found in multiple tissues. And the computer realized no matter which tissue came from, those are still B cells because the 20,000 numbers made the same pattern. Similarly for endothelial cells and similar for T cells. So that was like an early clue that this was all working, that we could analyze all these tissues and cell types that were similar but found in different tissues would give the similar sort of readout and different cell types would be viewed as different. One of the sort of metaphors we often use in the field for why do we spend all this effort to analyze individual cells is having to do with fruit smoothies. So you can imagine if you were trying to understand the difference between raspberries and blueberries and strawberries, and you were mixing them all together and turning them into a mixed fruit drink, it would be very hard to understand what each fruit was about individually. Whereas if you could analyze the pure fruit, that would provide much more direct information. And that is, in a sense, why we did this. Conventional approaches before this work involved taking entire tissues and analyzing all the molecules together. So all the cell types were mixed together, and it was hard to deconvolve them separately informatically. Whereas by working with each cell individually, it's sort of like having the ability to understand each fruit separately. So one example from this, here's the liver, and from the mouse data, looking at gene expression as a function of age, 1 month to 30 months. And you can see some genes have dramatic changes in their expression. These ones are selected for that purpose. And if you asked which genes were most changing with age, here's some that go up, here's some that go down. You could ask the number of cell types, which is like the number of fruits in the smoothie. Do they change? The number of cells change for a given cell type. Here's a cell type that doesn't change very much with age, but here's two that do change with age. And so you can ask this question very generally. Do changes in gene expression derive from the number of cells changing or from the intensity of expression in each cell? And that was a question that could never be worked out before this sort of work. And we asked it very generally. So we did the single cell measurements and we did bulk measurements, and we said, changes we see in the bulk measurements, how can they be explained from the single cell? Is it based on the effect size per cell, or is it based on the number of cells that appear? And in the vast majority of cases, it's both. So both contribute. Biology is very complicated in that respect. And you see, certainly in changes with age, often numbers of cell changing, but also intensity of gene expression per cell. And we'll return to this in the second lecture in a different context. Another thing we were able to look at was the structure of the immune system and the clonal relationship between B cells and T cells, which are the sort of key, foundational part of the adaptive immune system, and their clonality, which is to say their sequence relationship to each other. It changes over time. 3 months, 18 months, 24 months. You can see that the relationships between the antibodies and receptors they make are changing in time. This is an important part of aging, the change of the immune system with age. The whole third lecture will be about the immune system, and so this is a lead-in for that. And finally, the last thing I want to say on mouse, besides looking at natural aging, we looked at ways to reverse aging. And this has been, I mean, it was to my mind a controversial approach. And I was sure we were going to debunk the field, but we didn't. Turned out it was real. So the idea is something called parabiosis, which is Latin for living together. And if you are able to surgically attach two mice to each other so they share the same blood supply, and the blood goes back and forth between them, you can do very interesting experiments. And this had been a tool in the field of aging that Tony and other collaborators had used. And they had shown, the field had shown, that if you do parabiosis between a young mouse and an old mouse, so they share the same blood supply, the old mouse seems to have a reversal of aging and becomes younger in certain physiological effects. And this is something that has been parodied in the Hollywood movies and TV shows. If you watch the show Silicon Valley, there's stories of the executives trying to be younger by getting teenage blood infused. And as I say, I was very skeptical of this, but in fact, when we did the molecular analysis, it really is clear that there are interesting molecular changes that happen through parabiosis, and some of the effects we saw with natural aging can be reversed at the molecular level through this process. By mechanisms still as yet unknown. I'll just leave this one sort of figure up here as a result of a very complicated paper to illustrate sort of the power of taking an atlas approach. So here the question is, what gene expression changed with age and then were reversed by parabiosis? And we can ask that question in basically every cell type of the organism. So it lets you ask biological questions in a very interesting way. Previously, people had to ask their questions in one tissue or one cell type, and you always wondered, well, is that a general result or not? Is it specific to the tissue, or is that universal biological behavior? Here we can ask it across many tissues at once and understand which things are really universal and general and which are very specific to the biology of the tissue. And that is a great lens through which to appreciate biology. Okay, let's talk about human now. So we call this the tabula sapiens. We're kind of now in our sort of second generation of this. But in the initial one, we started with human organ donors, again, trying to get upwards of 24 tissues per donor, doing single-cell transcriptomics on all of them, but also doing conventional histopathology and also studying other aspects such as the microbiome. And again, a very large set of collaborators. I'll highlight just Bob Jones, who kind of quarterbacked the whole thing, but many other people contributed. We published the first version of this 3 years ago, and represented by this data. We've gone on to get more. And so the way to understand this, each row is a different tissue, each column is a different donor, and the size of the circle tells you the number of cells we obtained for that tissue from that donor. And initially, we had a few donors that had multiple tissues, but then we got to be very good at it over time, okay? And this doubled the size of the atlas and gave us much more of these sort of same donor tissues, which is a very powerful way to approach it. In mouse, we did it because of, I mentioned the limitation of the number of mice that could live that long. In humans, we wanted to do it because we wanted to have same genome in all the cells, same age, same environmental exposure, same epigenetic effects. I wanted to be able to have that constant across the experiment. So we're able to do that. How did we find humans willing to give us so many of their tissues? That came about through a unique and very special collaboration with an organization called Donor Network West. They're a nonprofit organization, and they serve Northern California and Nevada to help people who need organ transplants. And so in the United States, you can volunteer to be an organ donor. When you get your driver's license is when you sort of fill out the form to do that. In addition to giving your organs for transplant, you can also share them for scientific research. And so with appropriate consents and working with the group at Donor Network West, we were able to find donors and their families who agreed to let us use the tissues that were not being transplanted for scientific research. And it was a really sort of marvelous and amazing experience to work with them. We would often meet the families, which is very touching. You get a sense for the human aspect of this in ways that don't often come up in research. It was a lot of work to do these experiments. Typically what would happen is if there was a potential donor, we get a phone call from Donor Network West, and they'd say, we have a donor that may be, they're brain dead now, we maybe have them available for organ donations in about 3 days, have a look at their medical records, and we can consent the family. And so we'd look at that. If it looked like an appropriate one, we'd say, all right, this looks good. And if the family consented, we then would be able to have them donate their tissues. And the surgeons would be working through the night. This always seemed to happen at night for some reason. They'd be working through the night. Our folks would be waiting with ice buckets again. They'd be sent out to the labs and then all processed. And it was roughly 24 to 36 hours of nonstop work. I mean, really just absolutely challenging circumstances, incredibly dedicated students and postdocs and staff members and surgeons who would do this, and a unique sort of scientific opportunity. This is like at 5 in the morning, I think, and you can see there's exhausted people sitting in the hallways. And you can see from the masks, this was all done during the pandemic. We managed to keep it going during the pandemic. It was, again, sort of a testament to the dedication of all the folks involved, especially Bob, who was coordinating it all. And, you know, as I said, many, many people involved. This is sort of a list of some, but not all of them, but an incredible team effort. So let's return to some of the scientific results we got from this. Remember those transcription factors, the genes that discover other that control other genes, the second secret of life. In the human genome, there's about 1,622 of them that have been identified. And they haven't been identified, all of them experimentally. They've been identified by pattern matching and computational prediction. So these are predicted transcription factors in some cases. And many of them, their function is unknown. So we decided to try to study them all and see what we could learn. And in particular, one question we were interested in is the specificity. Is a transcription factor universal, expressed in all cells, or is it expressed only in one cell type, or only a few cell types? And we decided to try to answer that with our data. So there's a statistic you can compute called the tau factor, which sort of estimates this. This is the distribution of tau factors across all. We were able to measure the expression of 1,620 transcription factors. So all but 2 human transcription factors we've been able to find in which cell types they're expressed. And a tau factor of 1 means only in one cell type. Low tau factor means in many cell types. So you can see that there's many that are expressed in only a small number of cell types, and then a bunch that are expressed in many cell types. If you start to make figures of that, here's a graph. So each column here is a different transcription factor, and each row is the expression in a different cell type. The cell types are all color-coded. What I want you to take away from this is that there are many transcription factors that are expressed in just a small number of cell types. And so for those that have been unstudied as of yet, this tells you where to go to study their biology. So it's like a roadmap to understand the function of those transcription factors that have not yet been studied. For the ones that have been studied, we recover a lot of known biology. Ones in well-studied cell types, we see sets of transcription factors, many of which were known. It all sort of makes sense. And so for the understood biology, it makes sense, and this roadmap to new biology. Here are the 162 lowest tau transcription factors. So these are the ones that are most ubiquitously expressed. And so you can see that there are, by contrast, for a transcription factor that's expressed ubiquitously, has a very different pattern than the ones that are less ubiquitous. And if we focus in on those, the ones that are ubiquitous, so here it is now with order reversed. So this is transcription factors, all of them, in ascending tau order. You can see these are the ones that are highly ubiquitous. What are they? These are some of the gene names right here. And as we looked through them, we saw many that we recognized. Transcription factors that are known, used by all cells in terms of stress response, cell division and proliferation, metabolism and energy. That was great, that made sense. However, we also found some that were as yet unstudied. If you look at the ones that are ZNF, those are zinc fingers, and as you dig down into those are ones that are given a few letters in a name based on their putative structure, but essentially very little biology, essentially none known about them. And so amazing, for all the decades of studying cell biology, there are still transcription factors that are found in every single cell that are as yet uncharacterized. And so again, this points to some very interesting biology to follow up. We began to look at another phenomena in the tabula sapiens, which is related to aging, and that's called senescence. And there's something called the Hayflick limit that was discovered by Leonard Hayflick. And he discovered that cells are not immortal. They can only divide about 50 times before they stop dividing. And he called that physiology, that state when they stop dividing, he called that senescence. And based on this, he argued that there is a maximum human lifespan because you can only have so many cell divisions. And if you work out 50 cell divisions and the number of cells and such forth, upper limit of human lifespan is predicted to be about 120 years, and that's more or less what the record shows. Hayflick passed away last year, and someone at the New York Times had a lot of fun writing the headline for his obituary. Leonard Hayflick, who discovered why no one lives forever, dies at age 96. And he wrote a terrific book on aging as well. I didn't put it in here, but it's excellent. So we began to look for these senescent cells in our data in the tabula sapiens. And the field, generally speaking, has converged upon a definition of senescence, a molecular definition, which is expression of this gene, P16, it's also called TDKN2A, and not expressing this gene, MKI67, which has to do with cell division. So we looked for cells that had this RNA but not this. And when I say we, I mean Madhav Mantri, who's a postdoc in my lab. And through the entire tabula sapiens of 1 million cells, he found about 50,000 that fit this definition. And it's across the different tissues. Roughly, it's a couple of % of the cells, which is what you'd expect. And this turns out to be the largest set of human senescent cells from actual living human tissue that's ever been studied before. And the first thing we looked for was, what's the gene expression program for senescence? Is there are all senescent cells the same, or are there differences? And what we found is there's really no common gene expression program. I mean, if you looked at genes that are differentially expressed between senescent and non-senescent cells, and asked which cell types shared those differentially expressed genes, well, the only one that's in all of them is the one that defines senescence, CDKN2A. So that's like a bit tautological. But then even 2B and other things like IL-32 and IL-7, IL-6, not all cell types have that differentially expressed in senescence. And so there's a lot of heterogeneity in the gene expression program for senescence. And you can't just talk about a senescent cell as if they're all the same. But all is not lost. It's not total babble. We began to look for shared gene expression programs, and it roughly appears that there's about a dozen and a half gene expression programs that are shared, modules that are used across senescent cells, 6 broad classes of senescent cells, which you can see cluster by cell type, but not by tissue. So they're, again, found shared across different tissues, but they break out according to immune cells, endothelial cells, epithelial cells, stromal cells. And those cells appear to be senescent within a GLUT in a very similar way. And so there's broad similarities between some types of senescence, and we've started to articulate what that is. And it's, again, going to lead to, I think, some really fascinating insights into aging. So just sort of summarize where we are at this point. I hope I've convinced you that these large-scale atlases of single-cell transcriptomes are useful phenotypic companions to the genome, which provide a molecular definition of cell type and help interpret how the genome is used in different cell types. Now, I haven't talked at all about how this connects to conventional physiology of the cell types, because for hundreds of years, biologists would define cell types by their physiology. It turns out, we've done a bunch of work on this, and there's a whole other lecture I could give associated with that there's a remarkable relationship of physiology, of cell type and identity as defined by the transcriptome and as defined by physiology. It all kind of hangs together the way you hope it would if you believe the central dogma. So we, of course, aren't the only ones working in this field. There's been an enormous effort around the world using these approaches to study many different tissues in health and disease and across many different organisms. At CZI, we put together probably the largest database of everyone's data. So people now share their data, which is a wonderful thing. The tool is called Cell by Gene. You can go use it. You don't need to be an expert in bioinformatics. It's got a graphical user interface. You can ask it questions about your cell type or gene of interest. When I made this slide, there were 33 million cells in Cell by Gene. There's now 100 million cells. And as I had mentioned, the cost of getting this data has decreased by so much over the last decade that we recently announced a billion cell project. So we're going to increase by another order of magnitude the amount of data and fill in many of the sort of missing pieces, which includes many novel animals that haven't been studied yet. So how do you interpret all this data? How do you think about it? The tool we've arrived at for doing that is artificial intelligence and so-called large language models. And so I'll introduce that with one slide, and then we can jump into it. So we're in the midst of this amazing and scary technological revolution, right? I mean, if you played with things like ChatGPT or DALL-E or Claude or Mistral, which is the French version here, it's incredible. The tools to organize knowledge and information that one can derive from these approaches are phenomenal. And here's some examples where I was playing with a DALL-E, and I would ask it to make a picture of a family holding hands walking on the beach towards the sunset. Came up with this. Looks like a real picture. You can ask it to do a painting of all the proteins in a bacteria with DNA nearby in the Dutch master style. Came up with this. Or if you saw the poster for the talks, all the cell types in the human body in the impressionist style with a strand of DNA decorating the background. It comes up with this. Truly phenomenal. And it was able to do that because these models are trained on a huge corpus of image data and text data connecting that. And it's able to draw amazing conclusions and connections about the relationships between how we humans connect text and images together, and can generate things that don't appear yet in humans before they've done it in the world. So we've done exactly that with all the cell atlas data. We've trained a large language model on all that data. And the way you train a model in this field is to say you have a very complicated sort of numerical engine, essentially, and you show it some of the data of those 100 million cells. In this case, we used the smaller version, 36 million cells. So each cell, 20,000 dimensions. You cover up 20% of that data, and you ask the engine to predict it based on the rest of it. And you adjust, in this case, 650 million coefficients in order to get very good at predicting that data. And once you've done the best job you can predicting it, you have what turns out to be a very powerful model. And I'll say the other aspect of this that's very interesting for the experts in the audience, we did not need to use any of the genome data for these models. We used ESM2, which is a large protein language model, to embed each gene in a universal space there so that we could compare, we could train it on multiple organisms and test it on organisms it never saw before. So the whole model was designed from the beginning to be independent of any given genome or organism. And that was a very important philosophical choice. And a clever approach, I can't take credit for it, my collaborator Yuri Leskovich came up with that. So the model at the end of the day, 650 million parameters trained on 36 million cells by 24 GPUs over the course of a month and a half. And then with that model, we could put in all this annotated data that we had. And it's more than 1,000 cell types over 300 data sets, 50 different tissues, and 8 different species. So primarily human, followed by mouse, but also some other non-human primate, lemur, zebrafish, pig, a couple other non-human primates, and a frog. I mentioned Yuri Leskovich, a computer science professor at Stanford, who's my collaborator here, and we had 2 very talented students who did the work, Yusuf Rahani and Yane Rosa. So in building such a model, sort of the first thing you want to understand is how does it do on new data that is never seen before? Because you always worry about overfitting the model. You have a lot of parameters, and one sort of very important criterion for model performance is how does it do with new data it never saw before? So when we trained the model, we didn't give it Tabula sapiens 2 data. So that second batch of data from Tabula sapiens was unpublished. We didn't let anybody look at it or use it. The model was trained on all the other published data, including Tabula sapiens 1. But then after the model was built, we asked it, how does it do in understanding Tabula sapiens 2? So Tabula sapiens 1 here is shown embedded in blue and 2 in orange. And you can see they end up sitting right on top of each other. All the cell types from the new data that never seen before go right to the right place. They're closest to cells that are the same cell type from earlier data. So it does very well in held out data sets. How about if you give it a species that is never seen before? How can it do with that? How will it deal with that? So in this case, we again used held out data. This was published data from the green monkey, from the lymph node. That was not part of the training set, and nothing from green monkey was in the training data set. When we embedded it in the model and then asked what cell types is it closest to from other organisms, it does a pretty good job finding which are different cell types of the lymph node. These are all immune cells, and fibroblasts and endothelial. So it does a pretty good job figuring out cell types from organisms never seen before. Again, sort of an indication this might be a very useful model and a useful approach more generally. With all these models, we're very interested in emergent behavior. Like, can it learn things that wasn't really taught, that wasn't explicitly taught? So in this case, remember, it wasn't taught any biology. All it was given was a matrix of numbers. Cells, gene expression values, not names of genes, not biology of genes, not human annotation of cell types. So it was only trained on numbers. And then we can go back and ask, what happens to things where we know what the cell type is? And what's their distance in the embedding space at the end of the day? And so if you go through and ask within the embedding, how close are different types of T cells to each other? Well, it turns out they're pretty darn close. How close are other cell types of the immune system, like B cells? How about muscle cells, endothelial? And if you build this phylogeny of distances of these cell types in the embedding space, in the model space, it turns out they recapitulate what we understand about the biology of these cell types. So only from the numbers of mRNA molecules and nothing else, the model has learned some very interesting biology about the relationships between the cell types. And you can ask this question more generally as well. And so if you use the cell ontology tree, which is sort of a systematic way to characterize relationships between cell types based on their lineage, and ask parent-child, how close are they? This is the histogram of distances within the embedding space. Siblings, how close? That histogram. Cousins, second cousins, third cousins, so forth and so on. The more distant the relationship, the larger the distance in embedding space. And so it reflects, again, something very fundamental about cell lineage history and ontology. We've since gone on. This is called the universal cell embedding. At CZI, we built a second-generation model called transcriptomic, which has some more powerful aspects, such as it's generative, so you can predict, you can ask it to make you predict molecular profiles of cell types that aren't seen in nature, just like we did with the paintings at the beginning. That was just released on the preprint server last week, and the model for people to use. People are already using it. It's amazing how quickly this field is moving and how fast people take things up. So I can talk a bit more, or we can stop for questions. What's the custom?
It's usually an hour.
Okay, I could talk a bit more? Yeah.
You could carry on.
Okay, I'll carry on then.
Not for too long.
Not for too long. I'll finish on the hour. How about that? Okay. I do want to talk about some of the first health applications of these atlases. This is the work of a former student of mine, Sivan Verperian, where she wanted to understand cell type contributions to cell-free RNA. And so to get to that, I'm going to take a few minutes to explain to you what cell-free RNA is and how it works. So in your blood, there are molecules that derive from basically every tissue in your body. And that's true for nucleic acids as well. So there's DNA and RNA in the blood that are not inside of cells anymore. They're from cells that have died, lysed, or in the case of RNA, has been exported in some cases. And so there's a mixture of these molecules that would be nice to sort of deconvolve and sort out the contributions of which tissues and cell types are contributing to it, because maybe you'll learn something which cells are dying and which are diseased and so forth. This phenomenon of cell-free DNA and RNA is something that was actually discovered in France, in Strasbourg, in the late 1940s by Manuel Matthijs. And this was like before people even appreciated that DNA was the molecule of inheritance. I mean, it had just been worked out. Oswald Avery just published his experiments on that a year or two earlier. Very few people had read it. Even fewer believed it. These guys were just looking at blood chemistry from their perspective. And they were deciding to look for RNA and DNA, and they found it in blood. And this became the basis now of a very powerful set of diagnostics called liquid biopsies, once combined with things like sequencing and other clever ideas. This has been part of something I've been involved with for quite some time. We developed diagnostics to replace amniocentesis, for example, that are now used by millions of women around the world. And this will be the topic of the fourth lecture, and we'll dive into this in some great depth. But for the purposes of wrapping this one up, I want to return to the fruit smoothie analogy. Okay? So when you draw blood and there's cell-free RNA in the blood, that's the smoothie. Okay? So what can you tell about any particular contributor to that from the smoothie? Well, if you have the atlas and the molecular definition of each cell type, there ought to be a way mathematically to write this as a sum of all these. Okay? And that deconvolution that Sivan worked out and did a very nice job of that. I was able to, in fact, work out what the various contributions were of different cell types in the cell-free RNA. And you can see the largest contributions are from the immune system, not surprisingly, that had been appreciated. But many other cell types are contributing from many different tissues. And, you know, from the pancreas, from the kidney, from the large intestines, and so forth and so on. So in principle, you have, from a simple blood draw, signals of the health of almost every tissue in your body, including the brain, by the way. And so she was looking at data from, blood data from people with, in the one case, from the liver, fatty liver disease, and could see signals going up from liver cells. from her deconvolution. And in the case of kidney disease, a specific cell type, the proximal tubule cells, were decreasing in those, and they're known to be the ones that are destroyed in chronic kidney disease. So that reflected some of the underlying known pathology. So this was a very kind of nice first demonstration of how to use the cell atlas to understand sort of complex molecular signatures in the blood. The last example I'll give is one around therapeutic development, and it's actually an older story. It's something we did before we made the atlases, but is now an approach that's turbocharged from having the atlases. And it is to look at complexity of things like tumors and cancer. And Mike Clark and I had been working on that together for many years before, with the very earliest forms of these technologies, before we made the atlases, and had founded a company called Quanticell, which was trying to discover drug targets against the most sort of oncogenic subpopulations of cells within a tumor. And that ended up leading to 3 small molecule drugs, which all made it into the clinic in various stages of testing. And we'll see where they end up. But it's sort of pretty clear that you can use the single cell data to discover new drug targets where they're expressed in cases of the actual tumor, and also in cell types that you don't want them to be. So they would be rejected as a drug target if they're expressed in healthy tissues. And that's been a very sort of widely used use of the atlases to understand where potential drug targets are expressed in healthy cells where you don't want them to be. So let me wrap up here. We still have no way of predicting all the cell types of organism from the genome. Hopefully that'll come one day. Maybe based on the models we built on this experimental data. So we went out to experimentally characterize all the cell types of the, that's called the most common cell types in many organisms, and just provided a really interesting window into understanding how they arise, as exemplified by the story of the transcription factors. Building AI models, large language models on these data, provide a way to synthesize all the data into one reduced dimensionality space that crosses all of biology and evolutionary time. And these models are displaying very interesting emergent behavior with respect to cell biology. And again, as we'll touch on in future lectures, applications to human health are already well underway. So thank you.